So he's a German professor of biostatistics, epidemiology, and informatics at the UPenn Medical School. Hongzhe is actually very thoughtful, extremely thoughtful, and smart scholar. Every time I talk to him, I learn something from him. And he's one of the junior colleagues long time ago and became a leader and star in the field. And the reason I say that is because during his early career, I happened to write letters for his promotions or jobs hunting. So anyway, so he contributed a lot to large data analysis, particularly in genomic omic data. I forgot what is your, do you have a title for your talk?
Not projected.
Okay, so I think the instruction I got is, because we're already late, it will be 8 minutes talk, 5 minutes discussion.
I use this one. Okay. Thank you very much, Hejun, for the introduction. And I'd really like to thank the organizer for organizing this very nice retreat. And as you can see, some of the problem I'm going to talk about have very similar problem as the imaging problem in genomics. So first is there's a multi-modality data set. In genomics, for example, we have genomics, multi-omics, phenotypic data, social determinant of health, and this is the primary data. We also have ancillary data from other sources, such as phenotype data, clinical sequencing data, and the genetic testing facility data, single-cell data, and there are many other really complex phenotype data. It sounds very loud. And Xihong mentioned some of the large global data set. Can you hear me? Okay. No? Like a UK Biobank data, where we have a whole genome sequencing data for over 500,000 people. We have related EHR data, TopMed data set, and all of us data. Also, we have local data. For example, I work in the medical school. There's a Penn Medicine EHR data. We have Penn Medicine Biobank data. And what is this multi-modality data integration? Integration, you have to have a purpose. And I think one of the important goal of data integration is to understand the effect of genetic variation and the environmental factors on the biological processes and the complex phenotypes. And here, some of the machine learning idea I think could be very, very useful. For example, in the last couple of years, my group have been working on this idea of transfer learning. I just want to give you one example. So this is an example where we want to predict gene expression of one gene that belongs to a global central nervous system. And you can see, you can do the prediction in the tissue-specific way. You can use the data from one tissue, and you can also borrow information from many other tissues. And what you can see here, if you only use the data from one tissue, you can build a prediction model. But if you leverage data from other tissue, you can really improve the prediction performance. As you can see, the prediction error can reduce by over 20% by leveraging data from other tissue. So this is one example. Another example, you can also extend this transfer learning idea to the summary statistics-based transfer learning. For example, here at Penn, we have this Penn-owned biobank data. And we are interested in build a polygenic risk score for the lipid phenotype data for the Penn population, the population that related to Penn medicine. So here, as you can see, by using the summary statistics from UK Biobank, you can also improve the prediction R-squares. And this is another study that we published this year. We have a very nice cohort study called the CREEK study, where they look at the patients with chronic kidney disease. And you have this baseline proteomic measurement based on the blood sample. And you really want to understand their cardiovascular risk. So here, as you can see, uses proteomic data with some machine learning method, we can achieve much better time-dependent ROC curves than the standard clinical variable-based model. That's called the PCE. And we talk about this replication validation. We did try to validate our method, both use our own cohort and also use external other cohort. And you can see the performance still better than the clinical variable-based risk prediction model in the new cohort, but not as good as our own cohort, because those two cohorts, they have very different patient characteristics. So that's one of the problems we are really interested in, is how to do this domain adaptation in terms of building a risk prediction model. So for this integrative genomic, this is another one. Again, we talk about replication. This is a talk about the genetics of proteins based on our data. This is the estimated beta coefficient based on our cohort. And we try to replicate this result from two other published cohorts. One is a decode cohort, another is this interval study. And you can see the genetic effect on the protein. They are very similar, although the cohorts are so different. So I think we talk about this replication. This replication is extremely important. You can see that even the magnitude of the beta coefficient are quite similar. So this data quality is very high. This platform is a homologic platform. Is a very high quality proteomic measurement, and we are able to replicate them in multiple other cohorts. And to get this paper published in good journals, usually those replications are really important, are needed. And then you feel more comfortable, right, about the result if you can replicate all those results. So what is the next stage of research in this field? I had a very nice conversation with Kun Zhang at lunchtime. I think we need a new framework of causal inference and machine learning for this multimodal genomic data integration and modeling in order to understand the effect of genetic variation and the environmental factors on the biological process and the phenotypes. Some of the ideas from the recent machine learning AI field that could be useful, including the self-supervised learning idea to tackle the issue of you have small set of labeled samples, and also maybe some idea of transfer learning and use of synthetic data set. We also talk about the importance of explainability. You have to something you can understand the mechanism, right? You want to quantify the uncertainty. You want to have your model to have some interpretation in order to derive the causal and the mechanistic insight. And the other things is this low dimension representation idea. I think going to be very, very useful when you try to integrate multiple type of omics data. So how to develop some interpretable low dimension representation, I think is really important. Seems like Kun already worked this out, this problem. So this is one type of research I do, which I think the machine learning will play even more important role. Another type of data we work on is this microbiome metagenomic data. I just give you one example to give you a sense of the volume of the data. When you do the shotgun metagenomic sequencing for one single sample, you're going to have about 50 million very short sequencing reads. And each read is about 300 letters long, and they are derived from a mixture of hundreds of bacterial species. And to understand those sequencing reads, you also have to look at some of the database. For example, the reference bacterial genome database. In the current database, there are about 329,000 complete bacterial genome sequence, and each one is about 5 million base pair long. So it's a very large data set. And some really interesting features of such data is, for example, you can, when you study the microbial community, you need to look at the ecological relationship or phylogenetic relationship of the bacterial species. And after you align those data to the branches of the tree, you can think the data as a probability mass distribution on the branches of the tree. And for this type of data, for example, the graph neural network and this idea of optimal transport could be very relevant, very, very important. And another project we did is also interesting, basically just based on this cross-sectional shotgun metagenomic sequencing data, we figure out a way of estimating the bacterial growth rate, how fast the bacteria are replicating without observing the longitudinal data. It turns out this is related to the structured subspace estimation. So you have particular matrix with certain structure, but you have corrupted or permuted observation from that matrix with a noise, how to recover the underlying subspace. And the other one we did, this is my first try of using this recurrent neural network. I think it worked pretty well because this really allows us to incorporate many of the text information, the description of gene clusters and the similarity of the protein domains. And this recurrent neural network, I think, provide a really nice way of doing prediction. So for this particular project, we are interested in predicting the biosynthetic gene clusters in the bacterial genome sequence. We have a training data about 2,000 known biosynthetic gene clusters, and we want to identify all of them from the genomes of the bacteria. So for this microbiome field, I think the next stage of research is important to understand the function of the microbial genes, microbial variants. So she won't talk about human genetic variants. You can also look at the bacteria genes, bacteria genetic variants, and the gene clusters. What do they do? What are the functions of those genetic variants of microbial genomes? And also try to understand microbial functions by incorporating other type of data, such as metabolite data, host immune and gene expression data. Now, this is like Martin. I try to answer those questions. Prompts. What is the most exciting breakthrough that AI statistics can lead to? What will change the practice? One question I always have in my mind is when you have a lot of observational data, you have a small set of intervention perturbation data. Will those model work? How well those machine learning model works? For example, this transfer model, large language model works if you just put the observational data with interventional data together. And how this interventional data, small limited set of interventional data, can help you to train a better transformer model, for example. Then this is important. And what is the most important open question? This happened to be a problem where, so I'm working with GI doctors, and they have many years of EHR data on managing the IBD, inflammatory bowel disease. So we're talking about the possibility of building a personalized, not a treatment, personalized disease management, because there's no treatment for the inflammatory bowel disease. But there are over 10 different biologics, and whether we can build some learning algorithm to manage the disease. But it's very complicated. Some of you already mentioned the problem of missing data, some small sample size, also how to define the reward function, because different physicians, they score the treatment outcome very differently. But some of the idea of transfer learning, multi-arm band-aid could be useful for this problem. And I think the last one, and this is, I think many of you already mentioned this, what we should do. I think biostatisticians or statisticians should do first important scientific question. I can see when all the other speakers mentioned this one. The second one is this data engineering. We really need to be active involved in data engineering. For example, understand the pipeline flow of the data, manipulate the data, understand structured data, how to store the data. She who mentioned this, so she has the whole pipeline of doing this. And this is extremely important, because if you don't do this, you will not get to the key of the data, the issue of the data. And if you rely on other people to do this, they will lead the project, not you. And the other one also mentioned by Xihong is this computing environment. It's very important. For example, if you do the deep learning, how to do the GPU with optimized data access, data comments, cloud comments. Eventually, we do want to create a pipeline, a certain software, and we want to deploy those softwares by creating this user-friendly interface. I think this we should do more. That's it. Thank you.
Thank you, Hongzhe, for impressive work and also raising important point. I really feel bad that we already
Move
run
on.
over the time. So we don't actually have a question unless the boss give us extra time. But I guess we can save the question at the end, just in case we have time.
Yeah.
So now we go to the third speaker of the session. And so when you want, she's a faculty at Texas MD Anderson. And she received her PhD from Johns Hopkins.